So, good morning, students. My name is Ramanamurthy, Zoology faculty from Vijayawada. So, today we are going to discuss principles of inheritance and variations. That is a human genetics topic. And this human genetics topic is very crucial topic in NEET point of view. In final NEET examination, at least 3 to 4 questions will be given from this human genetics. So this human genetics includes several important areas like sex determination, next pedigree charts, pedigree analysis, and genetic disorders, 2 types of genetic disorders, that is Mendelian disorders and chromosomal disorders. So these are the important areas in human genetics. In final NEET examination, all types of questions will be given from this human genetics. Pedigree charts will be given, questions will be given from sex determination also, and also problems based on color blindness, hemophilia, sickle cell anemia, thalassemia, and based on chromosomal disorders, that is Down syndrome, Klinefelter syndrome. So diagrammatic questions also will be given in final NEET examination. So this is very crucial. This is concept-based topic. So when you understand the concept, then topic will be easy. So that's why concept-oriented topic. So you have to learn concept, then the topic will be easy. So first, what are the important areas in human genetics topic? First, sex determination. So while studying the human genetics, sex determination, who first discovered X chromosomes and Y chromosomes? X chromosome and Y chromosome. You know, X and Y chromosomes are called as allosomes. In human beings, 23 pairs of chromosomes present in each cell. Out of 23 pairs of chromosomes in each cell, 22 pairs, that means 44 chromosomes are autosomes. 1 pair are allosomes, that is sex chromosomes. So X and Y are the sex chromosomes in male human beings. XX are the sex chromosomes in female human beings. Sex chromosomes includes mostly X and Y. First, Henking was a scientist. When Henking studied the spermatogenesis in few insects means while studying spermatogenesis in a squash bug insect, when he studying the spermatogenesis in a squash bug insect, he identified the extranuclear structure in 50% of sperms in squash bug. That extranuclear structure in 50% of sperms in squash bug, that extranuclear structure was named as X body by the. So H. Henking identified extranuclear structure in 50% of sperms in some insects like a squash bug and named it as X body. Later, further investigations by some scientists like McClung. So McClung, while studying the spermatogenesis in grasshopper, he also identified another nuclear structure in 50% of sperms. And that extranuclear structure in 50% of sperms of grasshoppers is named it as X chromosome. So the name X chromosome was confirmed after further investigations by some scientists like McClung and other scientists. But Henking identified extranuclear structure in 50% of sperms while studying spermatogenesis in squash bug and named it as X body, not confirmed as X chromosome. Further investigations by McClung and other scientists confirmed that extranuclear structure in 50% of sperms in bugs and grasshoppers is named it as X chromosome. In some standard books, Henking discovered X body while studying spermatogenesis in firefly. So that is also given, but in our Chaitanya material, Henking discovered X body in a squash bug is given, but in some standard books, X body was discovered by the Henking while studying spermatogenesis in firefly is given. Both are correct. You should follow the same. Both are insects. But X chromosome that was confirmed by the other scientists like McClung while studying the spermatogenesis in grasshopper. This is very important. Next, Y chromosome. Y chromosome, and this Y chromosome was also discovered by another scientist like Stevens and Wilson. Stevens and Wilson, and these are the scientists discovered Y chromosome in a mealworm. In mealworm, scientific name Tenebrio molitor. So Tenebrio molitor is commonly called as mealworm, which is also a type of beetle. So in beetle mealworm Tenebrio molitor, Y chromosome was discovered by the Stevens and Wilson and also explained the role of Y chromosome in sex determination. So who first discovered X chromosome? That is X body by Henking, and that was confirmed as X chromosome by McClung and other scientists. Y chromosome was discovered by the Stevens and Wilson while studying spermatogenesis in mealworm. Scientific name is Tenebrio molitor, commonly called as beetle. So this is about the discovery of X chromosome and Y chromosome. Next, while studying genetics in sex determination, we should also follow the chromosomal or genetic sex determination. The sex determination by allosomes, allosomes means sex chromosomes, is called as chromosomal or genetic sex determination. In genetic sex determination, male heterogamety. So male heterogamety, we observed male heterogamety. So male heterogamety have two examples. That is, males are heterogametic. Heterogametic means they produces two types of gametes. Males are heterogametic, females are homogametic. So this male heterogamety we observed in case of grasshoppers, bugs. In grasshoppers, bugs, we observed XX and XO method of sex determination. That is example of male heterogamety. In human beings and Drosophila, we observed XX and XY method of sex determination. That is also example for male heterogamety. So this is given. This is a male human being and this is a female human being. What are the allosomes in male human beings are XY. What are the allosomes in female human beings are XX. So in male human beings, this is X chromosome and this is the Y chromosome. Generally, Y chromosome is short when compared to X chromosome and it is a hook-like structure. Next, one more important point. In case of male human beings, this Y chromosome is short, is a hook-like structure, and it also contains genes. The genes present on Y chromosome are called as Y-linked genes, also called as Hollandric genes. Hollandric genes. Hollandric means holo means complete, andric means males. The genes which are present on Y chromosome, the genes which are restricted to only males are called as Hollandric genes or Y-linked genes present on Y chromosome. In case of male human beings, X and Y are the allosomes. Y chromosome, Y chromosome is small when compared to X chromosome, and this Y chromosome contains genes called as holandric genes. And this Y chromosome in male human beings also contains another gene, that is TDF gene. TDF means testis determining factor gene is responsible for male fertility in human beings. TDF gene is the smallest gene in human beings. What is the smallest gene in human beings is TDF gene present on Y chromosome. And this TDF gene contains only 14 base pairs. Only 14 base pairs are present in TDF gene. TDF gene is the smallest gene present on Y chromosome. What is the largest gene in human beings? Dystrophin gene present on X chromosome. Largest gene present on X chromosome. Largest gene is dystrophin gene, that is given in molecular basis of inheritance, that is DNA fingerprinting and human genome project. Largest gene is dystrophin gene present on X chromosome. But what is the smallest gene is TDF gene present on Y chromosome. It contains only 14 base pairs. And Y chromosome also contains least number of genes, only 231 genes present on Y chromosome. This is also very important topic. So next, in human beings, what method of sex determination is? XX and XY method of sex determination. That is example of male heterogamity. And this is the Drosophila. In Drosophila also, what type of sex determination is XX and XY? Male Drosophila contains XY chromosome, and female Drosophila contains XX chromosomes. In male Drosophila also, this is X chromosome, Y chromosome is small and hook-like structure. But in case of Drosophila, according to Bridges, Y chromosome has no role in sex determination. Only sex chromosomes and autosomes play an important role in sex determination, according to Kelvin-Bridges genic balance theory. That point also you should remember. So, and another important point is, in case of Drosophila, male Drosophila is smaller and female Drosophila is larger. Male Drosophila is smaller and female Drosophila is larger. That is the important identification character between male Drosophila and female Drosophila. So that point you should observe. Next. So this point or that point is, in case of human beings and in case of Drosophila, XX and XY method of sex determination that we observe is example of male heterogamity. In case of bugs, grasshoppers and cockroaches, XX and XY method of sex determination, that is also example of male heterogamity. Next, in case of fowls, means birds, in case of reptiles, and in case of some fishes. In case of some fishes, what type of sex determination we observed is ZW, ZZ method of sex determination. So ZW, ZZ method of sex determination we observe in case of birds, that is fowls, reptiles and some fishes. In case of fowls, males shows AA, ZZ condition, and females contains AA, ZW condition. That is, males are homogametic and females are heterogametic. So birds, reptiles and fishes shows female heterogametic. That means females are heterogametic, males are homogametic. What are the allosomes? Allosomes in female fowls, ZW, male fowls, ZZ. Female heterogamety is also observed in some moths and some butterflies. And in moths and butterflies, what type of sex determination we observed is ZO, ZZ method of sex determination. So this ZO, ZZ method of sex determination we observed in case of moths and some butterflies. So this is also an example of female heterogamety. So example of male heterogamety is XX and XO method, XX and XY method. Example of female heterogamety is ZW, ZZ method, ZO, ZZ method. That is example of female heterogamety. So what about remaining sex determination that we discovered? We will discuss in another video. So next, after sex determination in fowls and human beings and moths. Next topic is sex determination in honeybees. Sex determination in honeybees is also an important. Sex determination in humans also the same. Human beings contains 23 pairs of chromosomes. Of 23 pairs, 22 pairs are autosomes and 1 pair is allosomes. So we will discuss in another short video. Next, an important area is sex determination in honeybees. In honeybees, scientific name of honeybee is Apis mellifera. So Apis mellifera is the scientific name of honeybee. So honeybees contains 3 types of caste, that is queen bee, drones and worker bees. In honeybees, what type of sex determination is haplodiploidy. So haplodiploidy type of sex determination. So this haplodiploidy type of sex determination observed not only in honeybees, it is observed in honeybees, also in ants, also observed in wasps. So in honeybees, ants and wasps, what type of sex determination is haplodiploid? Haplodiploidy means if organism contains haploid set of chromosomes, they are males. If organism contains diploid set of chromosomes, they are females. That is haplodiploidy, observed in honeybees, that is Apis mellifera, ants and also wasps. All these are belongs to an order, that is Hymenoptera. So this honeybees, ants and wasps belongs to which order is Hymenoptera. Hymenoptera members exhibit haplodiploidy condition. Haplodiploidy in honeybee includes queen bee. Queen bee contains diploid set of chromosomes. Diploid set of chromosomes in queen bees is 32 chromosomes. And also worker bees, they are also females, contains 32 chromosomes. But males are drones. Drones contains haploid set of chromosomes, 16 chromosomes. So if haploid set of chromosomes are present, they are males. Diploid set of chromosomes are present, females. What are female honeybees? Worker bees and queen bee. Queen bee is fertile. Queen bee is fertile female. Worker bee is sterile female. Drones are fertile males, contains only 16 chromosomes. So while studying the sex determination in honeybee, you should remember males means drones produces sperms by mitosis. They produces sperms by mitosis rather than meiosis. This is very important point. Which organisms produces sperms by mitosis? Male honeybees, that is drones. They are the fertile males. So male parent contains 16 chromosomes, sperms also contain 16 chromosomes. That means males produces sperms by mitosis. But queen bee, that is fertile female, produces ova by meiosis, that is by reductional division. This is very important. Next, another important point is, sex determination in honeybee. Drones do not have father, thus they cannot have sons, but they have grandfather and grandson. This is very, very important. So drones does not have father and sons, but they have grandfather and grandson. This statement is very, very important. So assertion reasoning question will be given. Assertion, drones does not have father and son, but they have grandfather and grandson. The reason is, drones are formed from unfertilized eggs, which are produced by the queen bee. Queen bee produced from the fertilized eggs. Answer, both A and R are true. R is correct explanation of it. So what is the reason drones does not have father and son, but they have grandfather and grandson? The reason is, drones are formed from unfertilized eggs. That's why they don't have father. But they have grandfather, because unfertilized eggs are produced by queen bee. Queen bee is produced from the fertilized eggs. That's why drones have no father and no son, but they have grandfather and grandson. So, for example, this is the unfertilized egg produced by the queen bee. From this unfertilized egg, drones are formed. The process of formation of drones from unfertilized eggs is called as male parthenogenesis, also called as archenotoky. Archenotoky, also called as male parthenogenesis. That means process of formation of drones from unfertilized eggs is called as male parthenogenesis, also called as archenotoky. So this is about the sex determination in honeybees. Haplodiploidy. Females contains diploid set of chromosomes. Males contains haploid set of chromosomes. And important point is, drones produces sperms by mitosis rather than meiosis. Worker bees does not produces ova because they are sterile females. That very important. They are also called as nurse workers. So this is the sex determination in honeybee. This is also very important area. Next important area is mutation. So mutation means, what is mutation? What is mutation means? Change in nitrogen base sequence of DNA. Mutation arises due to change in single base pair of DNA, and this is known as point mutation. This is very, very important. Change of single nitrogen base of DNA is called as mutation. Mutations occurs in sequence of DNA, also in structure of genes on chromosome. Two types of mutations: gene mutations, chromosomal mutations. Change of single nitrogen base of DNA is called as point mutation. A classical example of point mutation is sickle cell anemia. This is very, very important. What is the best example for point mutation is sickle cell anemia. Next, chromosomal mutations, chromosomal aberrations are commonly observed in cancer cells. The change in structure of genes on chromosome leads to cancers. That is mostly observed in cancer cells. So this is about the mutation, but in botany also you know about these mutations. Next important area in human genetics is genetic disorders. So in genetic disorders and pedigree analysis, this is an important area. Genetic disorders are mainly two types of genetic disorders. What are the two types of genetic disorders? Are Mendelian disorders. Second one is chromosomal disorders. These are the two types of genetic disorder. Genetic means they are either Mendelian disorders and chromosomal disorders. Gene disorders, Mendelian disorders also called as gene disorders. And second type of disorders are chromosomal disorders. What is the difference between Mendelian disorders and chromosomal disorders? Mendelian disorders or gene disorders are due to alteration of nitrogen based sequence of DNA. So due to alteration of nitrogen based sequence of DNA leads to Mendelian disorders or gene disorders. And these Mendelian disorders follow Mendelian pattern of inheritance. That is, they are inherited from one generation to another generation. Mendelian disorders. Chromosomal disorders. Chromosomal disorders are due to loss of one chromosome or gain of one chromosome. Gain or loss of chromosome in normal diploid set leads to chromosomal disorders. Chromosomal disorders are not inheritable from one generation to another generation. They are not heritable. But gene disorders, Mendelian disorders are inherited from one generation to another generation. But chromosomal disorders they are not transmitted from one generation to other generation. And one more important difference is Mendelian disorders exhibits pedigree analysis. Pedigree analysis is exhibited by Mendelian disorders or gene disorders. Chromosomal disorders does not exhibit pedigree analysis. So in Mendelian disorders, so we will discuss later about the pedigree analysis. First, Mendelian disorders. Mendelian disorders also called as gene disorders. Here this is the statement, very important. Genetic disorders may be grouped into two categories, Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by the alteration or mutation of single gene. These disorders are transmitted to the offsprings of same lines as we have studied in the principles of inheritance. The pattern of inheritance of such Mendelian disorders traced by the pedigree analysis. The pattern of inheritance of such Mendelian disorders can be traced in a family by pedigree analysis. So, pedigree analysis is exhibited by only Mendelian disorders, not chromosomal disorders. This point also very important. What is the most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria and thalassemia, etc. These are the most common and prevalent Mendelian disorders. Mendelian disorders are inherited from one generation to other generation, and they also follow the Mendelian pattern of inheritance. They also exhibit pedigree analysis. Most common is, you should remember, hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria and thalassemia. These are the most common and prevalent Mendelian disorders. So these Mendelian disorders also called as monogenic disease. They are also called as monogenic diseases, Mendelian disorders, because these Mendelian disorders are due to defect in a single gene. So due to defect in single gene, Mendelian disorders also called as monogenic diseases. Best example, sickle cell anemia, hemophilia, cystic fibrosis. All these are the best examples of the Mendelian disorders also called as monogenic diseases. Due to single gene defect, these disorders occurs in human beings. So students, so this is very, very important area. So what are the examples of Mendelian disorders? What are the examples of chromosomal disorders? So that you should remember, that is very careful. In Mendelian disorders, different types of Mendelian disorders. You know, what are the different types of Mendelian disorders are X-linked recessive, X-linked dominant, Y-linked, XY-linked, autosomal dominant, autosomal recessive. So there are different types of Mendelian disorders. So listen carefully. Mendelian disorders are again different types. X-linked recessive, X-linked dominant, Y-linked, XY-linked, autosomal dominant and autosomal recessive. These are the different types of Mendelian disorders, and these Mendelian disorders are inherited from one generation to other generation. So some of the best examples of Mendelian disorders, that is color blindness. Color blindness. Color blindness is an example of Mendelian disorder. Is a X-linked recessive, X-linked recessive gene disorder. X-linked recessive, also called as sex-linked recessive. X-linked, also called as sex-linked. X-linked recessive disorders, also called as sex-linked recessive disorders. And X-linked recessive disorders mostly follows crisscross inheritance. Mostly follow the crisscross inheritance. What is crisscross inheritance means? Transmission of a character or gene from father to grandson through carrier daughter is called as crisscross inheritance or zigzag inheritance or skip generation. That is mostly exhibited by X-linked recessive characters like color blindness, hemophilia, etc. So, in NEET examinations, color blindness is autosomal options. Color blindness is a autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. Answer is X-linked recessive or sex-linked recessive, whatever it is. Sex-linked recessive is correct, X-linked recessive is also correct. So generally X-linked recessive disorders are called as sex-linked recessive disorders. They follow the crisscross pattern of inheritance. So in color blindness, color blindness means the person unable to differentiate colors, mostly red and green colors. They are unable to differentiate. The most common type of color blindness is red-green color blindness, also called as daltonism. Red-green color blindness. This red-green color blindness is also called as daltonism. Daltonism, because the famous scientist Dalton also suffering from this disorder. That's why red-green color blindness is also called as daltonism. This red-green color blindness means the person unable to differentiate the colors, red color and green color. So this is very important, and generally X-linked recessive characters are more common in males, less common in females. This is very important statement. Generally, X-linked recessive characters like color blindness, hemophilia are more common in males, less common in females. This statement is very important. So this is the statement given. It occurs about 8% of males and 0.4% of females, color blindness. 8% of males, 0.4% of females. And this is because the genes that leads to red-green color blindness are on X chromosome. So this is the assertion reasoning question will be given in a NEET examination. Assertion. What is assertion? Red-green color blindness is more common in males, less common in females. Or red-green color blindness is observed 8% of males and 0.4% of females. That is assertion. Reason. The genes that leads to red-green color blindness are on the X chromosome. Both A and R are true. R is correct explanation. So question will be given in another way also. I already told. Suppose if you know the concept, then you can answer the question very easily. Suppose question will be given in different way. Assertion. Color blindness is more common in males, less common in females. That is assertion. Reason. Males are hemizygous for X-linked genes. Males are hemizygous for X-linked genes. Answer. Both A and R are true. R is correct explanation. Hemizygous. For X-linked genes, males are hemizygous. For X-linked genes, females are either homozygous or heterozygous. What is the meaning of hemizygous? occurrence of a gene in single copy in a diploid cell is called as hemizygous. So because of hemizygous condition, the color blindness is more common in males, less common in females. So this point is very important. So mostly regarding color blindness, they will be given problems based on color blindness. You know, protanopia. Protanopia is called as red color blindness. Deuteranopia is called as green color blindness. Red-green color blindness together called as daltonism. Tritanopia, another type of color blindness. Tritanopia. Tritanopia, and this tritanopia is also called as blue color blindness. Blue color blindness. But this is an example of autosomal recessive. Autosomal recessive gene disorder. Autosomal recessive gene disorder. Tritanopia. Color blindness is a autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, or sex-linked recessive. Answer is X-linked recessive or sex-linked recessive. That is correct. Suppose in examination specifically tritanopia is an example of is given, then answer is autosomal recessive gene disorder. Tritanopia means blue color blindness, but which is very rare. But most common type of color blindness is red-green color blindness. So that is very important. So in examination, what type of questions? If marriage happens to occur between normal woman and colorblind man, what is the probability of color blindness in their children? Suppose if marriage happens to occur between colorblind woman and normal man, what type of probability of color blindness in their child? Suppose if marriage happens to occur between carrier woman and normal man, then what is the probability of color blindness in their children? Suppose if marriage happens to occur between carrier woman and a colorblind man, so what type of probability of color blindness occurs in children? That type of questions will be given. Color blindness is very rare in females. This is very important. When colorblind girl is born, colorblind girl is born if both parents are colorblind, or if her mother is carrier and father is colorblind, then colorblind girl is produced. But color blindness is more common in males, less common in females. So students, that point is very important. So you should remember that points. Next, after color blindness, the most important, another one is hemophilia. So hemophilia is an also example of sex-linked recessive disease or X-linked recessive. Sex-linked also called as X-linked. Both color blindness and hemophilia, both are example of X-linked or sex-linked recessive gene disorders. What is hemophilia? Failure of blood clotting or delay of blood clotting is called as hemophilia. Hemophilia also called as bleeder's disease, which is also called as bleeder's disease, and it is first time observed in Queen Victoria royal family in England. So Queen Victoria royal family in England, this disease was first time observed. That's why hemophilia is also called as royal disease, because it is first time observed in Queen Victoria royal family. Failure of clotting of blood, delay in clotting of blood is called as hemophilia. So in hemophilia also there are three types. Hemophilia A, this is very important, hemophilia B, hemophilia C. There are three types of hemophilias. Most common type of hemophilia is hemophilia A, 80% of individuals. Hemophilia B, just 20% of individuals. Hemophilia C is very rare. Hemophilia C is very rare. Most common type is hemophilia A. 80% of individuals are suffering from this hemophilia A. Hemophilia A is due to deficiency of blood clotting factor 8. Hemophilia B is due to deficiency of blood clotting factor 9. Hemophilia C is due to deficiency of blood clotting factor 11. So hemophilia B is due to deficiency of factor hemophilia B is due to deficiency of factor 9. Factor 9, hemophilia B is mostly due to deficiency of factor 9, and this factor 9 also called as Christmas factor. This is also called as Christmas factor. So hemophilia B is due to deficiency of factor 9, and this factor 9 also called as Christmas factor, and hemophilia B is also called as Christmas disease, which is also called as Christmas disease, hemophilia B. So this is very important. Hemophilia B is also called as Christmas disease. Christmas disease is due to lack of factor 9. Hemophilia also follows crisscross pattern of inheritance. And this hemophilia is also more common in males, less common in females. And the possibility of female becoming hemophilic is extremely rare. This is assertion. The possibility of female becoming a hemophilic is extremely rare. That is assertion. Reason, because mother of such female has to be at least carrier and father should be hemophilic. This is the reason. So this type of question definitely will be given. So the possibility of female becoming hemophilic is extremely rare. This is assertion. Reason, suppose female is hemophilic, then her father is hemophilic and mother is at least carrier for hemophilia. So that's why if hemophilic daughter is present, definitely her father is hemophilic and mother is carrier for hemophilia. And hemophilic father is unviable in later stages of life. That is given. Unviable means, suppose if females have hemophilia, they will die by birth or by the age of 15 to 20 years they will die. Suppose if males have hemophilia, by the age of 35, 40 years, due to health problems, due to clotting defects, they will also die by the age of 35 to 40 years. That's why unviable is given in later stages of life. So this hemophilia is also lethal or dangerous in males. That's why hemophilic father is unviable in later stages of life. So this is about the hemophilia. Hemophilia is also an example of sex-linked recessive or X-linked recessive gene disorders. In our textbook, these two given for X-linked recessive or sex-linked recessive disorders. And X-linked recessive are more common in males, less common in females, and follow the crisscross pattern of inheritance. So next, students, another important topic is sickle cell anemia. This sickle cell anemia is example of autosomal recessive gene disorder. It is also called as SCD. SCD means sickle cell disease. Sickle cell disease or sickle cell anemia is an example of autosomal recessive Mendelian disorder or gene disorder. This sickle cell anemia is characterized by change of normal disc-shaped RBC into sickle shape. Due to conversion of normal disc-shaped RBC into sickle shape, the person is suffering from anemia. That's why this is called as sickle cell anemia or sickle cell disease. The sickle cell anemia is an example of point mutation. This is very important. This is an example of point mutation. This is best example of point mutation, and also example of codominance. It is also example of codominance. It is also an example of pleiotropy. pleiotropy, point mutation, codominance, pleiotropy, also example of autosomal recessive gene disorder, autosomal recessive gene disorder. So all these terms applicable for sickle cell anemia. Point mutation, best example for point mutation is sickle cell anemia disease. It is an autosomal recessive gene disorder. And also example of codominance, and also example of pleiotropy. You know pleiotropy. If one gene influences more than one phenotype, if one gene influences more than one character, that is called as pleiotropy. Example for pleiotropy: sickle cell anemia, phenylketonuria, cystic fibrosis. These are examples of the pleiotropy. So all these terms, point mutation, codominance, pleiotropy, autosomal recessive gene disorder, all these terms are applicable for sickle cell anemia. And one more important point you should remember regarding sickle cell anemia is it is associated with the 11th chromosome. Sickle cell anemia is associated with which chromosome is 11th chromosome. You should remember which chromosome is associated. And what are the important areas is the disease is controlled by a single pair of allele, that is HBA and HBS on 11th chromosome. So on 11th chromosome, for example, this is the 11th pair of chromosome. On 11th pair of chromosomes, suppose HBA is present, HBA is present, then that individuals are normal. HBA is the normal gene, HBA is the normal gene. On 11th chromosome, both the genes are normal, the persons are normal. Suppose on 11th chromosome of a person, on one chromosome HBA gene, on another chromosome HBS gene is present, then they are called as carriers or heterozygous. They are normal under normal conditions. They exhibit symptoms of sickle cell anemia only under low oxygen conditions or when they are reaching to high altitudes. Suppose on 11th chromosome, the person contains HBS gene and HBS gene. Suppose if both alleles are HBS on 11th chromosome, then the person is suffering from sickle cell anemia from childhood onwards, and they will die by the age of 15 to 20 years. So the severity is observed only in case of HBS. HBS persons are carriers but normal under normal conditions. HBA are normal. So how many pairs of alleles? A single pair of alleles, that is HBS. This HBS alleles are codominant alleles. This is very important. And another example of codominance: IA and IB alleles in ABO blood groups. IA and IB alleles in ABO blood groups also example of codominant. HBS alleles of sickle cell anemia also example of codominance. And out of this two alleles, total how many genotypes are possible? 3 genotypes. What are 3 genotypes? HBS, HBS. 3 types of genotypes are possible in sickle cell anemia. And the gene responsible for sickle cell anemia is HBS gene, that is formed due to mutation on 11th chromosome. This is very important. So what is the reason for sickle cell anemia? Is the defect caused by substitution of glutamic acid by valine at the 6th position of beta polypeptide chain. This is the main reason. The defect is caused due to the substitution of glutamic acid by valine. Substitution of glutamic acid by valine at the 6th position of beta polypeptide chain. Glutamic acid by valine at the 6th position of beta polypeptide chain. This is the main reason. for sickle cell anemia. Replacement of glutamic acid by valine at the 6th position of beta polypeptide chain. You know, hemoglobin consists of 2 alpha chains and 2 beta chains. Each alpha chain contains 141 amino acids. Each beta chain consists of 146 amino acids. Suppose when you observe the beta chain, of hemoglobin, in beta chain of hemoglobin, for normal HbA gene, what is the amino acid sequence is? First position is valine amino acid, second position histidine, third leucine, fourth threonine, fifth proline, sixth glutamic acid, seventh one is also glutamic acid. This is the amino acid sequence in beta chain for normal HbA gene. In the sickle cell HbS gene, that is mutated gene, what is the amino acid sequence in beta chain is? First one is valine, second histidine, third leucine, fourth threonine, fifth one proline, sixth one is, instead of glutamic acid, valine is present. So in normal gene, in beta polypeptide chain, 6th position is glutamic acid. In HbS gene, instead of glutamic acid, valine is present. So replacement of glutamic acid by valine at the 6th position of beta polypeptide chain is the main cause of sickle cell anemia. So why glutamic acid is replaced by the valine at 6th position? So this is the normal HbA gene. This is the coding strand. In coding strand, GAG is the sequence. This is the sequence GAG. On template strand, code is CTC on template strand. So on mRNA, what is the code is GAG. GAG is the triplet codon, code for which amino acid is glutamic acid. Suppose instead of adenine, thymine is present. So instead adenine is replaced by the thymine, that is instead of GAG, GTG is present in a normal coding strand. Then in a template strand, what is the sequence is CAC. On mRNA, what is the codon is GUG. GUG is the codon, code for which amino acid is valine. So due to change of triplet codon on mRNA from GAG to GUG. GAG is the triplet codon on mRNA. GUG is the triplet codon on mRNA in HbS gene. Then glutamic acid is replaced by the valine. That change of single nitrogen base, that is adenine, is replaced by the thymine. Change of single nitrogen base. Then glutamic acid is replaced by valine at 6th position. Then the normal disc-shaped RBC, this normal disc-shaped RBC converted into sickle shape like this. Due to conversion of normal disc-shaped RBC into sickle shape, the person suffering from anemia, and the disorder is called as sickle cell anemia. In sickle cell anemia, only HbS persons are severely affected. They will die by the age of 15 to 20 years. HbA persons are normal. HbS, that is carriers, they are normal under normal conditions. They have normal RBC under normal conditions also. When they face the problem of low oxygen conditions, then the mutated hemoglobin undergo polymerization that cause the change of RBC from disc shape to sickle shape. Then the person contains 2 types of RBC. So carriers contains 2 types of RBC under low oxygen conditions. They contains normal RBC and also sickle shaped RBC. But if they have sufficient oxygen levels. The RBC also normal, even though they have mutated gene in their RBC. So this is very important point. So assertion reasoning questions. Assertion reasoning question also will be given. So this is assertion question. The person with sickle cell anemia, in a person with the sickle cell anemia, the shape of RBC from biconcave disc to elongated sickle shaped structure under low oxygen condition. Person suffering from sickle cell anemia shows change of shape of RBC from biconcave disc to elongated sickle cell like structure. This is assertion is given. Reason, in sickle cell anemia persons, this is a reason. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension. This is a reason. Answer both A and R are true. R is correct explanation. So sickle cell anemia, regarding sickle cell anemia, you should remember it is an example of autosomal recessive gene disorder, example of codominance, example of pleiotropy, also example of point mutation. So these are very important regarding sickle cell anemia. The main reason is due to replacement of glutamic acid by valine at the 6th position of beta polypeptide chain. Students, this is very important. Next, phenylketonuria. It is also called as PKU, that is phenylketonuria, and it is a inborn error metabolism and associated with the 12th chromosome. It is associated with 12th chromosome. Sickle cell anemia is associated with 11th chromosome. And this phenylketonuria is also an example of autosomal recessive gene disorder. And this phenylketonuria is due to lack of an enzyme, phenylalanine hydroxylase. Phenylalanine hydroxylase is the enzyme that is deficient in phenylketonuria persons. What is the role of that enzyme? So phenylalanine, and this phenylalanine amino acid converted into tyrosine. It is converted into tyrosine in the presence of an enzyme, phenylalanine hydroxylase in liver. So generally phenylalanine is converted into tyrosine in the presence of an enzyme, phenylalanine hydroxylase. But in phenylketonuria persons, this phenylalanine hydroxylase enzyme is absent due to mutation of gene present on 12th chromosome. As a result, this phenylalanine is accumulated in brain and other cells of body. So phenylalanine or its derivatives, that is phenylpyruvic acid, phenylalanine accumulation or phenylpyruvic acid accumulation in brain and other tissues of body leads to a disease that is known as phenylketonuria. And excess of phenylalanine or phenylpyruvic acid excreted along with urine. That's why this disease was named as phenylketonuria, associated with 12th chromosome. It is also example of autosomal recessive gene disorder, also example of pleiotropy also. But it is a metabolic disorder. And what are the symptoms? Accumulation of this phenylpyruvic acid or phenylalanine in brain results in mental retardation or reduced skin pigmentation and hair pigmentation. Hypopigmentation of skin and hair. Hypopigmentation of skin and hair. That is also observed in phenylketonuria persons. So these are also excreted through the urine because of its poor absorption by kidney. So kidney, the nephrons of kidney unable to reabsorb phenylalanine. That's why it is passed along with urine. That condition is called as phenylketonuria. So this is also another important example. So next, another example is thalassemia. It is also an example of autosomal recessive gene disorder. So this is also an example of autosomal recessive. So sickle cell anemia is autosomal recessive. Phenylketonuria is also autosomal recessive gene disorder. Thalassemia is also autosomal recessive gene disorder. And this is also autosomal linked recessive blood disease transmitted from parents to offspring. So this is very important. Thalassemia is two types, alpha thalassemia and beta thalassemia. Alpha thalassemia associated with the 16th chromosome. This is very important. So alpha thalassemia is controlled by two genes present on the 16th chromosome. This is 16th chromosome. This is very, very important. And beta thalassemia associated with the 11th chromosome. So you should remember the chromosomes associated with the Mendelian disorders. Sickle cell anemia associated with 11th, phenylketonuria associated with the 12th, alpha thalassemia associated with the 16th, beta thalassemia associated with the 11th chromosome. So in alpha thalassemia, on 16th chromosome, this is the 16th chromosome. On 16th chromosome, two genes. What are the two genes is HBA1 and HBA2. HBA1 and HBA, two genes. Two genes means 4 alleles. But in NCERT textbook, what they have given is in alpha thalassemia, production of alpha globin chain is affected, while beta thalassemia, production of beta globin chain is affected. In alpha thalassemia, controlled by two close linked genes, HBA1 and HBA2 on chromosome 16th, and observed due to mutation is given. Mutation or deletion of one or more of the 4 genes. Four genes is given, but that is not correct. Actually, only 2 genes. 2 genes means how many alleles? 4 alleles. But you should follow. If same line will be given in a NEET examination, you should follow the same. That is also correct. Deletion or mutation of one or more of the 4 genes causes alpha thalassemia associated with the 16th chromosome. So 2 genes means HBA1, HBA2. Here also HBA1, HBA2. So total 4 alleles. Suppose if out of 4 alleles, one allele undergo mutation, becomes recessive, HB small a2. Person is suffering from 25% of alpha thalassemia. 2 alleles becomes recessive. Person is suffering from 50% of alpha thalassemia. 3 alleles become recessive, 75%. All 4 alleles recessive, 100% of alpha thalassemia. So in some children, only one allele undergo mutation and become recessive. That is 25% of thalassemia. In some children, out of 4 alleles, 2 alleles become recessive due to mutation. That is 50% of alpha thalassemia. 50% alpha thalassemia minor. Alpha thalassemia minor. 50%. That is not severe. Suppose if all 4 alleles are recessive due to mutation, that is called as alpha thalassemia major. That is very dangerous. That leads to death also. So major alpha thalassemia is dangerous. Minor alpha thalassemia is less severe, and child will survive and lead the normal life. So in alpha thalassemia, due to presence of recessive genes, there is no production of alpha chains of hemoglobin. You know, hemoglobin contains heme and globin. Globin contains 4 polypeptide chains, 2 alpha and 2 beta chains. So in alpha thalassemia persons, alpha chains are absent. Beta chains are present in hemoglobin or in RBC. In the same way, beta thalassemia associated with the 11th chromosome, but only one gene. In beta thalassemia, only one gene. This is the 11th chromosome. On 11th chromosome, HBB. This is HBB. So HBB, here also HBB. Suppose one allele undergo mutation, 50% of beta thalassemia. Two alleles, 100% of beta thalassemia. 50% beta thalassemia minor, 100% is beta thalassemia major. And beta thalassemia is also called as Cooley's anemia. It is also called as Cooley's anemia because discovered by scientist Cooley. So most common type of thalassemia is beta thalassemia. In beta thalassemia, beta polypeptide chains of hemoglobin are absent. Alpha chains present. In alpha thalassemia, alpha chains absent. In beta thalassemia, beta chains are absent. So this is very important. Thalassemia differs from sickle cell anemia in that former is quantitative problem. Former means thalassemia is a quantitative problem of synthesizing few globin molecules, while the later means sickle cell anemia is a qualitative problem. This is very, very important. Thalassemia is a quantitative problem of synthesizing few globin molecules, but sickle cell anemia is a qualitative problem due to synthesizing an incorrect functioning globin. That is the difference between thalassemia and sickle cell anemia. Thalassemia is a quantitative problem, sickle cell anemia is a qualitative problem. Similarity, thalassemia and sickle cell anemia both are blood related disorders and both are examples of autosomal recessive. What are the examples of blood related disorders of blood? Sickle cell anemia, thalassemia, hemophilia, all these are the disorders of blood. But hemophilia is example of sex-linked recessive or X-linked recessive. But thalassemia and sickle cell anemia are autosomal recessive. In sickle cell anemia is present. In thalassemia is also anemia is present. But thalassemia is a thalassemia is a quantitative problem. Sickle cell anemia is a qualitative problem. So this is about the autosomal recessive gene disorders. Best examples given in NCERT textbook: sickle cell anemia, thalassemia, phenylketonuria. Other autosomal disorders also there. So I will explain in next video. So next chromosomal disorders. So students, another important area is chromosomal disorders. You know, genetic disorders are two types. Mendelian disorders, also called as gene disorders. Second type is chromosomal disorders. Chromosomal disorders does not follow the Mendelian pattern of inheritance. They are not transmitted from one generation to another generation. That does not exhibit pedigree charts or pedigree analysis. Chromosomal disorders are mainly due to aneuploidy. Aneuploidy is the main reason for chromosome disorders in human beings. What is aneuploidy? Aneuploidy means it is due to non-disjunction. Aneuploidy is due to non-disjunction. What is non-disjunction? Failure of separation or segregation of chromosomes during anaphase stage of meiosis 1 and meiosis 2 is called as non-disjunction. Non-disjunction is the main reason for aneuploidy. Aneuploidy means either 2n plus 1 or 2n minus 1. This condition is called as aneuploidy. 2n plus 1 means, 2n means normal diploid set. Normal diploid set of chromosomes are 46 in human beings. 46 plus 1, that is contains 47 chromosomes in each cell. 2n minus 1, 2n means 46 minus 1, that is 45 chromosomes. 2n plus 1 or 2n minus 1 or 2n plus 2 or 2n minus 2, that is called as aneuploidy. The reason for aneuploidy is non-disjunction. What is non-disjunction? Failure of separation or segregation of chromosomes during anaphase stage of meiosis 1 and meiosis 2. Failure of segregation of chromosomes during meiosis 1 is called as primary non-disjunction. Failure of segregation of chromosomes during anaphase stage of meiosis 2 is called as secondary non-disjunction. So the main reason for aneuploidy in human beings is non-disjunction. What is aneuploidy? That is given, see, failure of segregation of chromatids during cell division cycle results in gain or loss of chromosomes. That is called as aneuploidy. Gain means 2n plus 1. That means instead of 46, the person have 47 chromosomes. Loss means instead of 46, they have 45. That is 2n minus 1. That is called as aneuploidy. That is the reason for chromosomal disorders. And example for aneuploidy is Down syndrome results in gain of extra copy of chromosome on 21st chromosome. Similarly, Turner syndrome results due to loss of next chromosome in human females. That means Turner syndrome persons contains 2n minus 1, that is monosomy. Instead of 46, Turner syndrome persons contains 45 chromosomes. And failure of cytokinesis after telophase stage of cell division results in an increase of whole set of chromosomes in organism, and this phenomenon is called as polyploidy. This condition often seen in plants. Polyploidy. Polyploidy is not a reason for chromosomal disorders in human beings. Polyploidy is mostly observed in plants. Polyploidy, suppose N means haploid, 2N means diploid, 3N, 4N, 5N. So this condition is called as polyploidy. Polyploidy condition is mostly observed in plants. What is polyploidy? Failure of cytokinesis after telophase stage of cell division results in increase of whole set of chromosomes. Whole set means total number of sets of chromosomes increases. That condition is called as polyploidy. So in 2024 NEET examination, so this question is given as statement 1 and statement 2. Statement 1, failure of segregation of chromatids during cell division results in increase of whole set of chromosomes. This is called as aneuploidy. That is given. Failure of segregation of chromosomes during cell division cycle results in increase of whole set of chromosome. This is called as aneuploidy. That is statement 1. Statement 2, failure of cytokinesis after anaphase stage of cell division resulting in gain or loss of chromosome, that is called as polyploidy is given. Answer is both statements are incorrect. Statement 1 is incorrect, statement 2 is also incorrect. What is polyploidy? Failure of cytokinesis after telophase stage of cell division resulting in increase of whole set of chromosomes, that is polyploidy. Failure of segregation of chromosomes or chromatids during cell division cycle results in gain or loss. That means at least one chromosome gain or one chromosome loss. That condition is called as aneuploidy. That resulting in chromosomal disorders like Down syndrome, Klinefelter syndrome and Turner syndrome. So what are the best examples? What are the best examples of chromosomal disorders is given? See, the first one is Down syndrome is given. Down syndrome is best example of 21st trisomy. 21st trisomy. And other name for Down syndrome is Mongolism. Mongolism is the other name for Down syndrome. Mongolism. It is due to trisomy of 21st. Down syndrome persons contains chromosomal composition of 45 autosomes plus XX or 45 plus XY. That means 45 autosomes, XX are the allosomes in females 45 autosomes plus XY are the allosomes in males. That means Down syndrome occurs either in males or in females, because it is a example of autosomal chromosomal disorder. Autosomal chromosomal disorder, because it is due to 21st trisome. 21st chromosome means autosome. Autosomal chromosomal disorders occurs either in males or females. Total number of chromosomes are 47. 47 chromosomes, either in male or female. 45 plus XX or 45 plus XY in males. So in males or in females, how many chromosomes are present is persons contains 47 chromosomes. Person suffering from Down syndrome contains 47 instead of 46. That is example of 2n plus 1, that is trisomy. It is explained by Langdon Down scientist. This is also very important. And Down syndrome persons show several important features, that is short stature, small round head, furrowed tongue, partially open mouth, palm is broad with the characteristic palm crease, physical, psychomotor and mental development is retarded. These are the features of the Down syndrome. So Down syndrome diagram is also given in NCERT textbook. You should follow this diagram. Some other symptoms also given, that is broad flat face, big and wrinkled tongue, congenital heart disease. That means Down syndrome persons also have heart diseases, palm crease, many loops on fingertips. So many loops means many lines on fingertips, flat back of head. These are the other symptoms of Down syndrome, also called as Mongolism, because people suffering from Down syndrome resemble Mongolian idiom. That's why Down syndrome is also called as Mongolism. Example of 21st trisomy, total number of chromosomes are 47, occurs in both males and females. Down syndrome persons are either male or female. They are fertile or maybe sterile also. Another important symptom of Down syndrome is furrowed tongue, partially open mouth, that is many loops on fingers, that is given. That is a fold is present on upper eyelid. That's why they cannot see straight, slant. Unusual eye fold is present in upper eyelid in Down syndrome persons also. Unusual upper eyelid, unusual fold on upper eyelid. So that is also another important symptom of Down syndrome. Next, Down syndrome is example of autosomal chromosomal disorder. Next one is Klinefelter syndrome. Klinefelter syndrome, and this Klinefelter syndrome was explained by scientist Klinefelter. Klinefelter syndrome associated with the 23rd trisomy. It is associated with the 23rd trisomy. Chromosomal composition is 44 plus XXY. That is total number of chromosomes 47. So it is due to 23rd trisomy. But Down syndrome is due to 21st trisomy. This is due to 23rd trisomy. 44 autosomes, but 3 allosomes, XXY. And Klinefelter syndrome persons are sterile males. It occurs only in males. Down syndrome occurs in both males and females. But Klinefelter syndrome occurs only in males. These are sterile males. And important features is an individual with Klinefelter syndrome has overall masculine development, feminine development, that is development of breast, gynecomastia is also expressed. Such individuals are sterile. That means unable to produce next generation. So gynecomastia, that is development of breast, one of the most important feature of the Klinefelter syndrome. And other important features of the Klinefelter syndrome is Klinefelter syndrome persons have tall stature and long arms and legs. Testes are degenerated. Testosterone content is very less. They are mentally retarded. These are the other symptoms of the Klinefelter syndrome. Example of 23rd trisomy. And 23rd trisomy means 2n plus 1. That is called as trisomy. Total number of chromosomes are 47. Turner syndrome. So Turner syndrome, this is example of 23rd monosomy. Example of 23rd monosomy. Monosomy means 2n minus 1. 2n minus 1 is called as monosomy. 2n plus 1 is called as trisomy. 2n minus 1 is called as monosomy. It is Turner syndrome is example of 23rd monosomy, and chromosomal composition is 44 plus x0. That means total number of chromosomes are 45. Only 45 chromosomes. Instead of 46, they have 45 chromosomes. Turner syndrome persons. Turner syndrome is explained by scientist Turner, and these are sterile females. Turner syndrome persons are sterile females. Klinefelter syndrome persons are sterile males. Down syndrome occurs both in males and females. Turner syndrome persons are also sterile females. Ovaries are rudimentary. Rudimentary means degenerated. Besides other features including lack of other secondary sexual characters. There is no secondary sexual characters. They have flat chest, short webbed neck, short stature, no female secondary sexual characters, and there is irregular menstrual cycles or lack of menstrual cycles. These are the other symptoms of the Turner syndrome. Chromosomal composition 44 plus XO. Total number of chromosomes are 45. Example of 23rd monosome. Klinefelter syndrome and Turner syndrome. These two are example of allosomal chromosomal disorders. Allosomal chromosomal disorders. That is defect in allosomes of chromosomes. That is either sex chromosome, one is lost, or gain of one extra allosome. But Down syndrome is an example of autosomal chromosomal disorder. Down syndrome is autosomal chromosomal disorder that is given in 2017 NEET exam. Which of the following is the autosomal chromosomal disorder occurs due to non-disjunction? Is Down syndrome. But Klinefelter syndrome, Turner syndrome, example of allosomal chromosomal disorder. They are also due to non-disjunction of chromosomes. So this is about the genetic disorders. Genetic disorders are two types: Mendelian disorders and chromosomal disorders. Mendelian disorders includes color blindness, hemophilia, sickle cell anemia, thalassemia, phenylketonuria. Chromosomal disorders, there are also two types: autosomal and allosomal. Autosomal chromosomal disorders are Down syndrome. Allosomal chromosomal disorders are Klinefelter syndrome, Turner syndrome. And the main reason for chromosomal disorders is non-disjunction of chromosomes that causes aneuploidy. Polyploidy has no effect in chromosomal disorders of human beings. So students, this is about the genetic disorders. Next, in this principles of inheritance, another important area is pedigree analysis. Pedigree analysis is also an important area. Symbols of pedigree charts also you should remember. Definitely questions will be given regarding pedigree analysis symbols. So what is pedigree analysis? What is pedigree analysis means? Pedigree analysis is a simple symbolic genetic diagram. It is a simple symbolic genetic diagram used to trace genetic disorders in human families. To trace the genetic disorders in human families, scientists discovered a simple symbolic genetic diagram that is called as pedigree chart or pedigree analysis. And this pedigree charts very useful identification of genetic disorders, Mendelian disorders like color blindness, hemophilia, sickle cell anemia in human families. And pedigree charts also very helpful whether the genetic disorder is dominant or recessive, or it is linked with X chromosome or Y chromosome, or linked with autosome. That is also we have to identify by using pedigree charts. So these are the pedigree charts. In pedigree charts, this is the male symbol, that is square. Next, this is circle is female. So darked symbols, that is darked circles, darked square, this is indicates affected. So suppose if there is only one line between male and female, only one line, that is mating between unrelated individuals. Suppose between male and female, two lines are present, that indicates mating between close relatives, that is called as consanguineous mating. This is very important. In 2022 NEET examination they have given. So if two lines are present between males and females, indicates mating between close relatives, blood relatives, that is consanguineous. What is the meaning of consanguineous? Means mating between blood relatives or close relatives. That is very important. Next, pedigree charts in last year 2025, this is the circle. In the center of circle, a dot that indicates carrier female, carrier female or heterozygous female, carrier or heterozygous female, carrier female or heterozygous female. So in case of X-linked recessive, this is the circle, middle of the circle a dot is present, that indicates female is carrier for X-linked recessive gene disorders like color blindness, hemophilia, that indicates. Suppose this is the circle, only half of the circle is dark and another half is empty, that indicates carrier female for autosomal recessive gene disorders. Autosomal recessive gene disorders. But carrier female for X-linked recessive is circle and middle dot. Suppose circle half is dark and half is empty, that is carrier female for autosomal recessive gene disorders. In case of autosomal recessive gene disorders, this is the square, half of the square is dark and half is empty. That is also indicates carrier male or heterozygous male. So these are the other symbols in pedigree charts. Next, in our NCERT textbook, one example is given for pedigree charts. What are the examples for pedigree chart is given is, so these are the two pedigree charts given in NCERT textbook for studying. So first one is example of autosomal dominant genetic disorder, autosomal dominant Mendelian disorder. So what is the best example for autosomal dominant is myotonic dystrophy associated with the 19th chromosome. So myotonic dystrophy is an example of autosomal dominant gene disorder. So how to solve this pedigree chart? So actually in pedigree chart, this is female is affected and male is normal is given. Female affected means in case of autosomal dominant gene disorders, suppose both alleles capital A are dominant or capital A small a, that indicates they are affected. In case of autosomal dominant gene disorders. Suppose both alleles are recessive, that is small a, both alleles are recessive, then the person is normal, not affected with disease. So this point you should remember in case of autosomal dominant. In case of autosomal recessive, if both alleles are recessive, small a, that is affected. In case of autosomal recessive. Suppose capital A, small a, or capital A, these are normal. In case of autosomal recessive. In case of autosomal dominant, capital A is affected, capital small a is affected, only small a is normal. But in case of autosomal recessive gene disorder, small a is affected, only both alleles are recessive affected. So capital A small a or capital A are normal. So these are very important. Suppose in case of autosomal dominant, here female is affected. Female is affected means you have to take capital A, small a. Male is normal, that is small a. Then possibilities is female is normal. Female is normal means small a. Here capital A, small a. Again here small a. Again here capital A, small a. So there are 4 children, 2 are females, 2 are males. 1 female, 1 male is affected. Affected persons have capital A, small a. Normal person, small a. Suppose if this affected female marries a normal male, again small a. Then they have 3 children, 2 males, 1 female. Males small a. Female affected, that is capital A, small a. This is the genotype. In case of male is marries another normal female. Suppose if male is marries another normal female, that is small a. So all are normal. No one is affected. This is an example of autosomal dominant is given NCERT, but this is also true for autosomal recessive. So this pedigree chart is applicable for both autosomal dominant and also for autosomal recessive. So don't be confused. Exactly is given, that is correct for both autosomal dominant and autosomal recessive. And this pedigree chart is exclusively for autosomal recessive. That is thalassemia, sickle cell anemia, phenylketonuria, etc. So I already said small a individuals are affected. Capital A, small a, capital A is normal in case of autosomal recessive gene disorders. Autosomal recessive gene disorders, here both male and female are normal is given. Normal is given, but you have to take their carriers, that is capital A, small a. The possibility of offspring, here male is normal. Normal means that is capital A. Here maybe male is affected. Affected means small a. Here capital A, small a. Here again capital A, small a. Here small a. Here capital A, small a. So what are the genotypes of offspring is normal capital A, affected small a. And again female not affected capital A, small a. Affected female small a. Normal male capital A, small a. Suppose if this normal female marries another normal male, then they have 3 children, 2 daughters, 1 son. Son is affected, that means small a. And here capital A, small a, maybe capital A. This is the genotypes of the offsprings. In pedigree charts, this is the first generation. You should remember. This is the first generation, and this is the second generation, and this is the third gen. Generations indicates with the Roman numbers. Offsprings are indicates with the normal numbers 1, 2, 3, 4, 5. Generations are indicates with the Roman numbers 1, 2, 3. So this is about the pedigree charts. Pedigree charts will be given in another way. X-linked recessive, X-linked dominant, Y-linked pedigree charts also will be given. But in NCERT textbook, only 2 examples is given. That is autosomal dominant and autosomal recessive. This is autosomal recessive. This is autosomal dominant, and this is also applicable for autosomal recessive. Students, this is about the pedigree charts. You should practice pedigree charts, then definitely will be perfect. So, students, these are the sum of questions given in previous NEET examination. That is in 2021 NEET examination, 2022, 2023, 2024. Some questions I have given. See, this is the first question. Which one of the following conditions correctly describes the manner of determining the sex in a given example? So, homozygous sex chromosomes determine male sex in Drosophila. Actually, in Drosophila, sex of the offspring is determined by the males, that is XY chromosomes. Homozygous sex chromosomes determine the female sex in birds. Homozygous sex chromosomes, that is not ZZ, ZW chromosomes of females determine the sex of the offspring in case of birds. XO type of sex chromosomes determine the male sex in grasshopper. So this is the answer. In case of grasshoppers, what type of sex determination is XX and XO method. So XO type of sex chromosomes determine the male sex in grasshopper. So this is the right answer for this question. XO condition in human as found in turners determine the female sex. No. So that is also wrong. Next, second question. So this is the diagram for some Mendelian disorders. So suppose if female mother is affected, the genes are transmitted to son. If father is affected, the genes are transmitted to daughter. So this type of Mendelian pattern of inheritance is observed in which Mendelian disorder? That is in hemophilia. It's an example of X-linked recessive. Sickle cell anemia, thalassemia, phenylketonuria, all these are autosomal recessive. Autosomal recessive disorders are transmitted to both males and females, but from mother to son, father to daughter, that is in case of hemophilia. So this is the second question. Third question is a normal vision man whose father was colorblind marries a woman whose father was also colorblind. They have their first child as daughter. What are the chances of that this child would be colorblind is 0%. So there is no chance of daughter as colorblind. In case of normal vision man whose father was colorblind marries a woman whose father was also colorblind. That means she is carrier. So from carrier mother, the gene is transmitted to son, not to the daughter. That's why answer is 0%. Next, another question. Pick out the correct statements. Hemophilia is a sex-linked recessive disease, correct. Down syndrome is due to aneuploid, is correct. Phenylketonuria is an example of autosomal recessive gene disorder, correct. Sickle cell anemia is an X-linked recessive. This is wrong statement. So which statements are correct for this question is A, B, C are correct. This is wrong. Sickle cell anemia is also example of autosomal recessive, not X-linked recessive. So next, another question. So match the following. Column 1 and column 2, dominance, codominance, pleiotropy and polygenic inheritance. Dominance means in heterozygous organism only one allele express itself, that is dominance. Codominance, in heterozygous organism both alleles express themselves fully, that is codominance. Pleiotropy, a single gene influences many characters. Polygenic inheritance, many genes govern a single character. So A2, B3, A2, B3, C4 and D1. So this is the right answer for this question. So next, a disease caused by autosomal primary non-disjunction. So already I told just now, example for autosomal primary non-disjunction disorder is Down syndrome. So Down syndrome is autosomal primary non-disjunction. Klinefelter syndrome is due to allosomal primary non-disjunction. Turner syndrome is also allosomal primary non-disjunction. Sickle cell anemia is an example of Mendelian disorder. So next, thalassemia and sickle cell anemia are caused due to a problem in globin molecule synthesis. Select the correct statement. This also just now we discussed. This is 2017 NEET examination question. So thalassemia is due to less synthesis of globin molecules. This is the right answer. Thalassemia is due to less synthesis of globin molecule. Already I told this is thalassemia is a quantitative problem of synthesis of few globin molecules, but sickle cell anemia is a qualitative problem. Here, fourth option, sickle cell anemia is due to quantitative problem is given. That is wrong statement. Both are due to quantitative defect. That is wrong statement. Both are due to qualitative. That is also wrong statement. Next, in a marriage between male with blood group A and female with blood group B. So male with blood group A, female with blood group B. The progeny had either blood group AB or B. What could be the possible genotypes of the parents is generally the parents have this type, that is IA, IO and IB, IB. So suppose if parents have IA, IO and IB, IB genotypes, their children have either AB or B blood group. So this is the another question. Match the items of the column 1 with column 2. This is another match the following question. So this is also a previous NEET examination question. XX and XO measure of sex determination. XX and XO measure of sex determination observed in grasshopper, and XX and XY measure of sex determination. XX and XY measure of sex determination means female homogamety. Karyotype 45, that is Turner syndrome. ZW, ZZ measure of sex determination, that is female heterogamety. So answer is A3, B, B4 and C1 and D2. D2. So this is the answer, right answer for this question. Next, assertion reasoning question. Down syndrome, Klinefelter syndrome and Turner syndrome are chromosomal disorders. This is given in AIMS, previous AIMS examination, 2019. And Down syndrome, Klinefelter syndrome and Turner syndrome are chromosome disorders. Correct assertion. In Klinefelter syndrome, female or sterile, that is wrong. That is Klinefelter syndrome, male is sterile, but here female or sterile is given. So A is true, R is false. Next, what is the genetic disorder in which an individual has an overall masculine development, gynecomastia, and is sterile? So gynecomastia, I already told, gynecomastia is the most important symptom of Klinefelter syndrome. So the right answer is Klinefelter syndrome. Next, select the correct match, that is phenylketonuria. It is autosomal dominant. That is wrong. It is autosomal recessive. Sickle cell anemia, autosomal recessive. This is correct. Associated with the chromosome 11. Thalassemia, X-linked wrong. Hemophilia, Y-linked wrong. So the right answer is sickle cell anemia is autosomal recessive trait associated with chromosome 11. Next, match list 1 with list 2. Hemophilia. You know, hemophilia is a disorder of blood, delay of clotting of blood. It is due to the sex-linked recessive disorder, defect in blood coagulation. A2, Down syndrome. Down syndrome is additional copy of chromosome number 21, phenylketonuria. This is inborn error metabolism which lacks an enzyme that converts phenylalanine to tyrosine. Klinefelter syndrome. Klinefelter syndrome is presence of additional copy of X chromosome. So the right answer is A, A2 and B4, A2, B4, C1 and D3. D3. So second option is right answer for this question. Next, assertion is in a question. A father will never pass the gene for hemophilia to his son. This is correct. Father always transmit the hemophilic gene or colorblind gene to his daughter, not to sons. Reason, hemophilia is a sex-linked recessive trait or X-linked recessive trait. So answer is both A and R are true. R is correct explanation. It's a correct explanation. So this is also a very important question. Next, which of the following statements are correct about Klinefelter syndrome? So statements, this disorder was first described by Langdon Down. So spelling is missed. That is Langdon Down. No, Klinefelter syndrome was discovered by explained by the Klinefelter. Langdon Down explained Down syndrome. Such an individual has overall masculine development. However, the feminine development is also expressed. That is correct. The affected individual is short stature, no, long stature. They are very tall, more than 6 feet or 7 feet tall. Physical, psychomotor and mental development is retarded. That is for Down syndrome. And such individuals are sterile. So which statements are correct for Klinefelter syndrome is? So B statement is correct and E statement is correct. So B and E only. So these B and E statements are correct for Klinefelter syndrome. Next, broad palm with single palm increase is visible in person suffering from Down syndrome. This is most important symptom of Down syndrome, also called as Mongolism. Next, which one of the following symbols represents mating between relatives in humans? So mating between relatives means consanguineous marriage. That is mating between close relatives. So which symbol indicates consanguineous means? This is the symbol which indicates consanguineous marriage, that is mating between close relatives. This is a previous NEET question 2023. Next, in a cross between a male and female, both are heterozygous for sickle cell anemia gene, what percentage of progeny will be deceased? Is 25%. This is also previous NEET question in 2021 or 2022 NEET examination, previous NEET question. So very important type of question. So in both parents, both male and female parents are heterozygous for sickle cell anemia. So for example, for this type of questions, you have to take female is heterozygous, that is capital A, small a. Male parent is also heterozygous, that is capital A, small a. That is heterozygous, that is carriers. So then what percentage of progeny will be deceased? Then you have to take a simple capital A, capital A, capital A, small a, capital A, small a, and small a, small a. So out of 4 children, capital A is normal, capital A, small a is carrier, that is normal, capital A, small a is carrier, normal. Only small a persons are affected. So out of 4 children, only 1 children is affected, that is 25% of children are affected. So this type of questions will be given in 2026 NEET examination also. So you should be very careful. So next, the trait traced in this pedigree chart. This is an example of pedigree chart. So you should follow as an example of autosomal recessive gene disorder. Next, assertion. In a person affected with sickle cell anemia, shape of RBC changes from biconcave disc to elongated sickle cell structure under low oxygen. Reason, in sickle cell anemia, the mutant hemoglobin molecule undergoes polymerization under low oxygen tension. So this is a assertion reasoning question. Answer is both A and R are true. R is correct explanation. Why normal disc-shaped RBC turns to sickle shape? Due to mutant hemoglobin molecule undergoes polymerization under low oxygen tension. That is the correct reason. That's why answer is both A and R are true. R is correct explanation for this question. So last question, if both parents are carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in affected child? Same model. So for any autosomal recessive gene disorder, whether it is thalassemia or phenylketonuria or sickle cell anemia, whatever it is. So suppose if both parents are carriers, just now I told, if both parents are carriers means capital A, small a, male parent is also carrier, that is capital A, small a. That is capital A, capital A, capital A, small a, capital A, small a, and small a, small a. That means out of 4 child, only 1 child, this child is affected. 4 child, 1 child means 25%. So for autosomal recessive gene disorders like thalassemia, sickle cell anemia, phenylketonuria, affected child contains both alleles recessive, that is small a, small a. That you have to consider. Suppose if both alleles are dominant or one allele is dominant, one allele is recessive, you cannot consider they are affected. If both alleles are recessive, they are affected. And in this question, how much percentage of child affected is only 25%. That is, out of 4 children, only 1 children is affected, that is 25%. So this type of questions definitely will be given. So you should practice. So good luck to all of you. Thank you.